Welcome to G-Fun Facts Online. Is your mind just a ghost in the machine, a clever illusion, or is it a computational process, a language spoken by the very matter of your brain? This idea, called the language of thought hypothesis, suggests that thinking is like computation, manipulating mental symbols according to a kind of grammar. For decades, we thought this language was exclusive to our biological neurons. We built silicon imitations, artificial neural networks, but they were always just that, imitations. Today, we're on the verge of a revolution. Scientists are no longer just imitating neurons. They're building them from the ground up, using the most versatile material in existence, proteins. We're going to explore how these molecular machines are learning to speak the language of thought and what it means for the future of intelligence itself. To understand how radical it is to build a neuron from scratch, we first have to appreciate the original. The biological neuron is the core unit of your nervous system. Think of it as a tiny information processor. It has a cell body, the soma, which is its factory. Branching out are dendrites, which are like antennas, receiving signals from thousands of other neurons. And then there's a long cable, the axon, which transmits signals away. The language of a neuron is both electrical and chemical. Inside a single neuron, communication is electrical. It uses a signal called an action potential, a rapid all-or-nothing electrical spike that travels down the axon. This happens when the neuron receives enough excitatory signals at its dendrites. These signals open ion channels, letting positive ions flow in. If the internal charge reaches a certain threshold, it triggers a chain reaction, and the neuron fires. But between neurons, the communication is chemical. The connection point is a tiny gap called a synapse. When an action potential reaches the end of an axon, it releases chemical messengers called neurotransmitters. These molecules float across the synapse and bind to receptors on the next neuron's dendrites. This binding can either excite the next neuron, making it more likely to fire, or inhibit it, making it less likely. Your brain is performing this intricate dance of electrical spikes and chemical signals billions of times a second across trillions of connections. It's the foundation of every thought, feeling, and action. Inspired by this biological marvel, computer scientists created the artificial neuron. The classic model is the perceptron. It's a mathematical function that mimics the real thing. It receives numerical inputs, and each input is given a weight, a number representing its importance, much like the strength of a synapse. The neuron calculates the weighted sum of these inputs. This sum is then passed through an activation function. If the sum exceeds a certain threshold, the neuron fires and produces an output, like a 1. If not, it stays inactive, outputting a 0. An artificial neural network, or ANN, is just a collection of these simple units arranged in layers. The real magic is how they learn. During training, you show the network examples, like pictures labeled cat or dog. At first, its guesses are random, but by comparing its output to the correct answer, you can calculate an error. This error is then used to slightly adjust all the weights in the network, a process called backpropagation. After millions of examples, the network fine-tunes itself to recognize patterns. But let's be clear, ANNs are not brains. Your brain has 86 billion neurons with trillions of connections. The largest ANNs are vastly simpler, and while a silicon chip is fast, the brain is orders of magnitude more power efficient. Your brain does its incredible work on about 20 watts of power, less than a dim light bulb. Training a large AI model can consume enough energy to power a small town. Biological neurons use spikes, while many artificial ones use continuous values. Our brains learn through complex processes we still don't fully understand, while ANNs use well-defined math. ANNs are powerful tools, but they are a simplified abstraction. This gap between silicon and biology is what's driving scientists to a new frontier, to a new substrate for thought: proteins. Proteins are the workhorses of life. They're built from just 20 types of amino acids, but the sequence determines how a protein folds into a unique 3D structure, and that structure defines its function. This inherent programmability makes them the perfect candidate for building artificial neurons. Three key properties are at the heart of this new endeavor. First is molecular recognition. Proteins can bind to other molecules with incredible specificity. Like a key fitting into a lock, an antibody finds its specific virus. An enzyme finds its specific substrate. For a protein-based neuron, this could be the input mechanism. Instead of numerical inputs, it could directly sense the concentration of specific molecules like neurotransmitters or drugs. The strength of this binding could act as the weight in an artificial network. Second, conformational changes. Proteins aren't rigid. When a molecule binds to a protein, the protein can change its shape. This change can act like a molecular switch. We can engineer a protein so that in one shape it's off, and when an input molecule binds, it switches to an on state. This is a direct physical analog to a neuron's activation function. The summation of input signals, molecules binding, causes a state change that produces an output, like activating an enzyme. Third is self-assembly. Nature doesn't use factories to build complex structures. It uses self-assembly. You can design individual proteins to spontaneously connect in a pre-programmed way, forming larger architectures like fibers or cages. This means you could design protein neurons that automatically wire themselves into a complex network, building a circuit from the bottom up. These three properties, recognition, switching, and self-assembly, provide a powerful toolkit for building the next generation of computing devices. And this isn't just theory. Scientists are already building them. Deep in oxygen-starved mud, a bacterium named Geobacter sulfuridescens has a strange ability. It can breathe minerals. It does this by extending tiny, electrically conductive protein filaments called nanowires. For years, scientists were baffled by how a protein structure could conduct electricity. It turns out the secret is in their structure. These nanowires are formed by proteins stacking themselves in a way that creates a continuous path for electrons to flow, just like in a copper wire. A team at the University of Massachusetts Amherst, led by Jun Yao, harnessed this natural wonder. They created a simple artificial neuron using these bacterial nanowires. They built a device called a memristor, whose resistance changes based on the charge that is passed through it. The crucial breakthrough was that these protein nanowires allow the device to operate at incredibly low voltages, the same 60 to 100 millivolt range as our own biological neurons. Previous artificial neurons required far too much power to ever interact with living cells. By integrating this nanowire memristor into a circuit, they created a device that perfectly mimics the leaky integrate and fire behavior of a biological neuron. The circuit slowly accumulates charge, and when it hits a threshold, it fires a voltage spike, then resets. It generates repeatable action potentials, just like a real neuron. Even more impressively, they showed it could respond to the neurotransmitter dopamine, proving it can speak the chemical language of the brain. While the Geobacter approach is a bioelectronic hybrid, another team at Caltech, led by Michael Elowitz, is building computational circuits entirely inside living cells. They developed a synthetic protein circuit they call a perceptine, a mashup of perceptron and protein. This circuit, built inside mammalian cells, performs a type of neural network computation known as winner-take-all classification. The perceptine is a masterpiece of protein engineering. It computes a weighted sum of molecular inputs using reversible protein binding. It then uses engineered viral proteases, enzymes that cut other proteins, to perform activation and inhibition. When one part of the circuit, a node, receives the strongest combined input signal, its protease turns on. This protease then does two things. It cuts and inactivates the proteases of all the other nodes, and it cuts a tag off of itself that would have marked it for destruction. This creates a powerful feedback loop. The strongest node becomes more stable and actively suppresses all its competitors. It's the winner that takes all. This entire system, starting with just 8 types of proteins, generates a complex network of over 300 chemical reactions. The Caltech team showed this protein neural network could accurately classify the levels of different signals inside a living cell, with a decision boundary that could be tuned. These two examples, the nanowire memristor and the perceptine, are profound proofs of principle. They show that the fundamental operations of a neuron can be implemented using proteins. We are opening the door to a new form of biocomputation, one not just inspired by life, but built from it. But a single neuron isn't a mind. The real power is in the network. So what are the grand challenges on the path from a protein neuron to a thinking machine? First is scalability and stability. How do we go from one neuron to millions? For the nanowires, it's a manufacturing problem of placing them precisely on a chip. For the perceptines, the complexity inside a cell grows exponentially, putting a huge metabolic load on the host cell and risking unpredictable side reactions. Then there's the interface problem. How do you wire up a protein? The nanowire is conductive, which is a direct link. But for chemical circuits like the perceptine, reading the output often means waiting for a fluorescent protein to glow, which is slow and indirect. We need high-bandwidth interfaces between the chemical world of proteins and the electronic world of our computers. And if we ever want to use these devices inside the body, they must be biocompatible. The immune system is designed to attack foreign proteins. Engineering proteins that can fly under the immune system's radar is a monumental challenge. Beyond these technical hurdles lies the deepest question of all. Could a system built from proteins ever truly think? Could it be a physical home for the language of thought? Let's revisit that idea. The philosopher Jerry Fodor argued that thought requires a system of symbols and a syntax for combining them. A protein network might just fit the bill. In this system, a symbol wouldn't be a 1 or a 0, but a specific molecule. The representation for cat might be the presence of a particular signaling molecule or the shape of a specific receptor protein. This syntax or the rules for combining these symbols would be the laws of chemistry and the logic of the engineered protein interactions. The thought John loves Mary could be a physical reality, a molecular assembly where the John molecule and the Mary molecule bind to a scaffold, which then allows the loves molecule to attach. The resulting complex is the physical manifestation of the thought. The logic of the perceptine, with its irreversible cuts, is a perfect example of implementing rule-based operations at the molecular level. A system like this could also be productive and systematic, able to generate a near-infinite number of thoughts from a finite set of concepts. The combinatorial possibilities of molecular assembly are vast. If your system has the parts and rules to assemble John loves Mary, it inherently has the parts and rules to assemble Mary loves John. The capacity for complex thought would be built directly into its molecular hardware. This is, of course, a huge speculative leap. But what these protein-based neurons give us is a tantalizing glimpse of how the abstract architecture of thought could be implemented in a substrate other than our own brain. It suggests mentalese might not be exclusive to our particular brand of biological hardware. It could be a general property of any system with enough symbolic and syntactic complexity. The road ahead is long, but the potential is transformative. In the near term, we could see revolutionary smart therapeutics. Imagine engineered cells with perceptine circuits that analyze disease markers in your body and release a drug only at the precise time and place it's needed. Or biosensors that interface directly with our nervous system, giving early warnings for neurological disorders. In the longer term, the dream of brain-inspired neuromorphic computing could be realized in its most literal form. We could build computers whose fundamental processors are protein molecules, devices that would be incredibly power efficient and biocompatible, leading to seamless brain-machine interfaces that could restore lost function or even augment our own minds. Ultimately, the quest to build a mind from molecules is more than an engineering project. It's a philosophical journey. It forces us to confront the deepest questions about what we are. What is the relationship between mind and matter? By learning to speak the language of proteins, we may finally begin to understand the language of thought itself. We may be witnessing the dawn of a new age of intelligence, one that is not born of silicon and electricity, but folded from the very amino acids that are the alphabet of life. Thanks for listening to G Fun Facts Online.